afternoon, everyone. Today we are going to have a 30-minute lecture for our course Neurodevelopment, and I will be talking about adult neurogenesis. We started our course by talking about embryogenesis, and from there on we talked about neural development in the embryonic stages, and in the last weeks we talked about migration and neurite growth, and finally in our last week we will be looking into neural development in the adult brain. So in our previous lecture where we talked about the migration, we gave the example of cerebral cortex development in telencephalon, and we saw that everything starts with the ventricular zone at the prosencephalon stage, where we have the radial glia-like cells that act as both the neural progenitor cells and also the ladders that differentiating neurons use to migrate. And the first thing we will observe after the ventricular zone, the first layer that forms by differentiation from the ventricular zone is the preplate. And we know that preplate then separates into two different layers: the marginal zone that contains Cajal residue cells and the subplate. And the cortical layers are formed by differentiating cells of the ventricular zone. These cells will migrate between marginal zone and subplate, and then they will start to stack there on top of each other in an inside-out manner to form the cortical plates. So around these stages where the cortical plates are being formed, we will also see the development of another layer which is called the subventricular zone. And the subventricular zone carries similar characteristics with the ventricular zone. And we finished our previous lecture by saying that the subventricular zone is the only remnant of proliferating cells in the adult brain. So these ventricular zone structures, when we look into the adult brain, they will be forming the neuroepithelial cells that form the walls of the lateral ventricles, in general ventricles, and the subventricular zone will persist to have neural progenitor cells in the adult human brain. So I will start from there. I will start talking by these cells, these neurogenic, so-called neurogenic niches in the adult brain. So by definition, adult neurogenesis means the formation, the production of new neurons in the adult organism, in the adult brain. And it is a unique form of structural and functional plasticity, which I will elaborate also in the following time. And we consider it as a continuum of embryonic neurogenesis. However, it carries different characteristics. Both the cells of the subventricular zone and the function of these differentiating cells differ quite a lot compared to the embryonic development. Let's just review these processes once again. We know that there are three important processes during neurogenesis. The first one is the proliferation, and during proliferation we can see both symmetrical cell division and asymmetrical cell division. Symmetrical cell division will be used as a way for proliferation, for self-renewal of the particular cell type, whereas asymmetric cell division will serve as a source of differentiation, as you know. So the main type of neural progenitor cells in these adult neurogenic niches, we will see that they are again radial glial-like cells. And these cells are usually called either type 1 cells or the major neural progenitor cells. We will see that through a series of asymmetrical cell division, these cells can either be forming new astrocytes, so we can see gliogenesis, or they can differentiate into a type of neural progenitor cell that proliferate a lot, that divide very fast, and then they serve as a transient step in the differentiation of neurons, which are called transiently amplifying cells. As the name implies, these cells will divide a lot, but they will not stay alive for a long time, and in the end they will either differentiate or die. So remember that we talked about apoptosis a little bit, programmed cell death, and apoptosis is a process that occurs in parallel to both proliferation and differentiation. So at each of these steps, we can also observe programmed cell death. And especially the transient amplifying cells have a higher apoptosis rate, because as I said, for instance, we might have 4 transiently amplifying cells, most likely only one of them will differentiate into neuroblast and move on to the next stage, but rest of these cells will die. The next steps in the differentiation processes following with the major neuron is the neuroblast. And as we see differentiation at each of these steps, we can also say that, as we saw earlier, with each differentiation, these neural progenitor cells become more specialized, so they will lose their self-renewable capacity a little bit, and also they will become less potent in terms of differentiation. So the most specialized type of neural progenitor cell will be the neuroblasts, and this is the last step before actual neuron. And the neuroblasts usually differentiate and migrate at the same time, or they might differentiate into an immature neuron, and that immature neuron might migrate. So also in the adult brain, the last step will be the migration and the following maturation of that immature neuron. And during maturation, of course, we have some preliminary steps. First of all, we need neurite growth, which will be axonogenesis and dendrogenesis. We need the formation of new synapses, which will be synaptogenesis. And then, after obtaining enough amount of inputs, both excitatory and inhibitory, the neuron will finally mature and become a fully mature newborn neuron. So this scheme, this process is observed in all of the neurogenic niches. So we will have neural progenitor cells that have the highest capacity of self-renewal and potency, but then they will go through a series of differentiation, and then we will end up with the immature neuron that needs to gather enough inputs to mature. So let's have a look at the neurogenic niches. In the adult brain, we don't see neurogenesis everywhere, and we can already guess that because we know that the brain is a very elegant structure that has many finely defined connections. So having a high amount of neurogenesis, having a high amount of newborn neurons is actually not a very good thing. So we need to control it, and we will see that the neurogenic niches exist in only very small portions, very well-defined borders, within very well-defined borders in the brain. And adult neurogenesis throughout the last 2 or 3 decades, actually we observed adult neurogenesis in different regions, but the later studies defined them either as constitutive or reactive, meaning that either we have an ongoing neurogenesis in that particular region, or we observe neurogenesis in response to an injury, for instance. When we look into neocortex, both in the rodent brain and the human brain, normally we don't observe neurogenesis there in the postnatal organisms, in the adult organisms. However, as a response to an injury, we might observe that some astrocytes actually differentiate into neurons, which will more likely be interneurons, not excitatory pyramidal neurons of the cortex. However, as I said, this is reactive, and there is actually many reports against that kind of neurogenesis in the adult brain. The next place we can see is, as we saw, the subventricular zone. Subventricular zone neurogenesis is actually quite pronounced in the rodent brain. However, when we look into the human brain, we see that it's almost diminished. The main reason is thought to be functional. The subventricular zone, the neurons that differentiate from the subventricular zone will become the neurons in the olfactory bulb, and we know that the olfactory bulb constitutes a larger portion of the brain in rodents compared to humans. We have an almost diminished structure compared to rodents. And also we don't depend on olfactory cues as much as, for instance, rats do. Olfaction is not our primary sense. Therefore, the requirements of neurogenesis seems to be less in some of the mammals. including humans. So subventricular zone is a very important neurogenic niche in some of the mammals, but for humans we actually have neurogenesis there only in the first year of life. Then we have dentate gyrus of hippocampus. Hippocampus, as you would know, is a very important organizer in the long-term memory, and it is always very active in all of the mammals. It's a very important structure. Dentate gyrus also has a prominent role in the trisynaptic loop of hippocampus. It's a very active place. And in dentate gyrus, under the granule cell layer, we have a structure called subgranular zone, and it also acts as a neurogenic niche. Now, since it is active in, for instance, both rodents and the humans, we would expect adult neurogenesis to continue there even in humans, and this is actually what we observe. However, I will actually talk about this a little more in a few minutes. Even in dentate gyrus, after some years, after actually when the adolescence is done and when the organism moves, when the human moves to become a mature adult, we actually see again diminishing adult neurogenesis even in dentate gyrus. Also, striatum is an interesting place as a neurogenic niche. In the rodents, we have few reports in favor of also neurogenesis in the striatum. However, with humans, it seems to be compensating at least a little bit for the lack of subventricular zone neurogenesis. Also, we can talk about hypothalamus maybe, but hypothalamic neurogenesis is only observed in the very early months of the rodent brain, early months, postnatal months, and it is not very pronounced in the human brain. So let's look at the structure of subventricular zone. Subventricular zone is located on the walls of lateral ventricle, and it consists of these radial glial-like cells, which are also called type B cells. So when we look into the differentiating cells, we actually see again radial glial-like cells being the most important source for both proliferation and differentiation. Then we have populations of transiently amplifying cells and neuroblasts. So in the rodent brain, as you would see here, subventricular zone will be located towards the striatal side of the lateral ventricle, and in the human brain the location is exactly the same. The structure of subventricular zone differs a little bit between rodents and humans, and the main reason is actually in the adult organism, in the adult human, the structure changes a little bit because there isn't a pronounced neurogenesis there anymore. So one interesting property of subventricular zone neurogenesis is that the source of new neurons is quite far away from actual target olfactory bulb. So these cells will need to migrate a long way, and we see a migration route here, which is also called RMS or rostral migratory stream. And when the neuroblasts are differentiated, they will actually take this route and go to olfactory bulb cortex and then they will, when they reach to their final destination, they will actually mature there and be integrated into the neural network. So this is a section from rat brain, and it is a staining against glial fibrillary acidic protein. And glial fibrillary acidic protein, or GFAP shortly, is a biomarker for astrocytes. And as we know, radial glial cells are a subtype of astrocytes. So you can see the ventricular space here, and on the walls of the ventricles you see a very dense layer that is stained that is GFAP positive. And if we look closely, we will see these cells that have very long extensions and that have almost an oval-like cell body. And these are the type B cells. These are the radial glial-like cells. Some of these cells will reach to the veins, the capillaries, and well, they will function as normal astrocytes, whereas some of them will continue to proliferate, and then some of them might undergo a series of differentiation. They will differentiate first into type C cells, which are the transiently amplifying cells, and these type C cells will be located around the type B cells, and they will immediately differentiate, as I'd say, to the type A cells or the neuroblasts, and then these neuroblasts will have to migrate to their final destination. So this is a summary of the structure of subventricular zone. If you look at subgranular zone, we will see that it is, as I said, located immediately underneath the granule cell layer, and in these pictures we see a double staining by hematoxylin, which stains all the cell bodies, and we see again GFAP staining. So here you might see these purple layer, which is the granule neuron cell layer, and immediately underneath that you will notice that there are again these long extensions of radial glial cells and the cell bodies located here. So the extensions of radial glial cells this time extend into granule cell layer, and these cells will differentiate also into granule neurons. So the main difference between subgranular zone and subventricular zone neurogenesis is that the differentiated cells don't have a long distance to migrate this time. They will differentiate here in this zone, and they will just go up into the granule cell layer. So they have a much shorter distance of migration. And again we see that radial glial-like cells are the main proliferating and differentiating cell population here. They are also called type 1 cells. Then we have the transiently amplifying cells, which are called the type 2 cells. Then we have neuroblasts that are called type 3 cells. And we will see that when the neuroblast is finally differentiated into an immature neuron, this immature neuron will then vertically migrate into the granule cell population immediately above subgranular zone, and then there we will see neurite growth and synaptogenesis, and when there is enough amount of inputs, both excitatory and inhibitory, these cells will mature and they will be integrated into the neural network of hippocampus. And this is, we can say that this is a summary of the structure for subgranular zone. Now, at this point, before I will not go in too much detail with the structure and the cell types, but I want to present another perspective, an evolutionary perspective. So we know that we already saw that actually this neurogenesis is a very complex process. It has at least 3 steps of differentiation, and it is controlled by very strictly controlled by many different cues, including both the chemical cues and the physical cues by the extracellular matrix. So it's a very complex process. It requires a very intricate control and regulation. It is energetically very expensive, and also it bears certain risks, like every proliferating cell pool also has the probability to become a tumor, cancerous tumor. And we know that actually most of the brain tumors are formed from the astrocytes, and they become gliomas. So the neuroblasts basically have also carry the risk of becoming a tumor-like structure in the end. So there is also a risk of containing neurogenic niches. So the question rises then, why do we have at all neurogenesis in the adult brain? Why don't we just use the neurons that were already formed? As you would know, neurons cannot divide. So maybe we can think about this. The first thing we would think is if something happens to a tissue, if there is a loss of neurons, this is the only way to compensate. However, we know that adult neurogenesis is actually not really regenerative, because it is very limited to certain locations, and its capacity is not actually enough to compensate for the cell loss in a certain region. It is actually, adult neurogenesis seems to be a functional, seems to have a functional purpose. It's a unique form of plasticity, as I said before. So it looks like it's for the especially important places, it seems to be increasing plasticity capacity actually. So this looks like its main purpose. So we have some proofs for why adult neurogenesis might continue, but the thing is, we can also ask, how did it evolve? And we can also ask what is similar to and what is different from other species as well. There are many different answers to these questions, but I will just talk about the phylogenetical reduction a little bit. So when we look into embryonic neurogenesis versus adult neurogenesis, we see that there are some capacity differences, cell type differences, functional differences also. And as I said, regenerative neurogenesis and adult neurogenesis are exactly, they are not the same thing. And the adult neurogenesis that we see in mammals today doesn't seem to contribute to regeneration, but it seems to be more involved in a type of plasticity. And we see a low amount of neurogenesis, so there might be some other questions that we can ask, like are low levels of neurogenesis still functionally relevant, or are there vestigial quiescent remnants of stem cell niches, and can these be reactivated in some way? So this second question is much more interesting in terms of using this capacity as a therapeutical target. And we know that there is proof that even low number of newborn neurons can be highly functional, can increase the capacity, plastic capacity of a certain region. So yes, the first question, are low numbers of neurogenesis functionally relevant? Actually, we know that answer is yes. I will then use my rest of my time to introduce some differences between different animal species. We know that there is a strong reduction of brain neurogenic and reparative capacity from fishes, amphibians, reptilians to mammals. There's a strong reduction in adult neurogenesis and brain repair. And when we look into mammals only, we see that even from rodents to humans, there is a very strong reduction. And we can also ask, why do we have that reduction? And we can first, for instance, focus on subventricular zone. So when we compare, one question would be, is it because our lifespans? So a mouse would generally, on average, live for a year or maximum a few years. On the other hand, humans can live up to 70 to 90 years. So the time that passes is much larger. Maybe if we weren't, looking into the human brain after all these years, we would still see some neurogenesis. This doesn't seem to be the case, because we look at dolphins that have a lifespan of 40 to 50 years, and there is no neurogenesis in their brain at all, even in the very early postnatal days, in the earlier time of their life. So it seems to be, it seems to have a different reason. And as I said again, subventricular zone produces olfactory neurons, and when we look at the use of olfaction, yes, mice depend on it more. Humans, well, our olfactory bulb is much smaller and we don't depend on that as much, but dolphins don't use it at all. Dolphins don't have olfactory system like the terrestrial animals, because they live in water, they're aquatic mammals. And since there is no use of this, it seems like it's fully functional. Since there is no use for new neurons, olfactory neurons, well, it seems like there is no neurogenesis in subventricular zone. Here we see DCX positive cells in subventricular zone. DCX double cortin is a microtubule associated protein. We use it as a biomarker of neuroblasts and immature neurons. So this is the staining from postnatal humans, and when we look at the brain after 1 week after birth, we see actually that there are lots of neuroblasts, lots of DCX positive cells, and the number declines quite a lot when we come to first month. In 6 months, we have a very low amount of neuroblasts, and when we come to 12 months, there is almost no neuroblasts left, only we see a few. And in 18 months, no neuroblasts at all. So we lose subventricular zone neurogenesis in the first year of our life. So as I said, the subventricular zone neurogenesis seems to be more related, the strong reduction seems to be more related about the function of olfaction bulb. Alright, so let's move on and let's look at other studies. Now we see the number of proliferating cells and their change by age in mice and humans, we used Ki67, which is a nucleic antigen. We use it to detect proliferating cells, and we say that every month the number of proliferating cells, every month after birth, the number of proliferating cells decrease sharply, even for mice. And when we look at 40-year-old humans, well, we will see that there is a very little number of proliferating cells, and specifically in the subgranular zone. Now we are talking about subgranular zone. When we look at cetaceans, which is the aquatic mammals, we see in the subgranular zone, we don't see any double cortin positive cells at all. And when we look at bats, for instance, these are 4 different species of bats, we see very low numbers of Ki67 positive cells, and again we see very low numbers of DCX positive cells. So the hippocampal neurogenesis also, similar to subventricular zone neurogenesis, it seems to be also diminishing in other types of mammals. If you look at humans, human subgranular zone, immediately after birth, we will see a very high DCX positive, very high number of DCX positive cells. After 3 weeks, we would see less, and as we go to 1 year, around 1 year, we will see that it is we have a lower number of neuroblasts in the subgranular zone. However, we can still detect new neuroblasts up to 13 years, and if we go to the adulthood, around 35 years or 70 years, we wouldn't expect to see neuroblasts at all. So subgranular zone neurogenesis persists for a longer time. However, it also diminishes in adulthood in humans. So this is just another way to show this. So these are the regions, dentate gyrus, in different species. So this is for the rat, this is for human, and this is for dolphin. And as I said, in different species, even for the subgranular zone, which is a very active brain region, it still diminishes in adulthood in most of the mammals. All right, so let's think a little bit about the function of hippocampus as well. Hippocampal network, together with the entorhinal cortex, is the main region for spatial memory. Long-term memory, yes, but more importantly, spatial memory, which is basically forming a map of our physical environment. And when we look at these animals, yes, all animals depend on this. However, we might think that maybe terrestrial animals are depending on that more, especially when we look into animals that live in complex underground structures like the rats. That might also explain why we have different levels of neurogenesis in even subgranular zone. And also remember that olfactory bulb is a very important cue to locate spaces, to locate physical environments for rats. So it seems to be neurogenesis, adult neurogenesis in rats or rodents seems to be helping with the long-term memory. However, it is, especially for spatial memory, however, it is decreasing in different mammals and by adulthood. So there are still a lot of questions that we can ask. The first thing is, can we, of course, from a therapeutical approach, can we reactivate this? Is there any way to reactivate these cells? Are they still there or are they gone for good? This is one interesting question. Also, we study with lab animals, and they may not entirely reflect the wildlife animals. So this is also a question that we might ask. And the question seems to be also related to the species, their environment, the modality, density, and complexity of the environmental stimuli, and the workload of these particular regions. So we know, at the moment, of course, we know a lot compared to a few decades before about adult neurogenesis. However, there are still lots of questions that we can ask, and this is why it is still a very interesting and a very fun area to work with. and we need more multidisciplinary, we will need more multidisciplinary studies on this topic. Out-of-the-box thinking always helps, and if you are interested in that kind of study, this is what I'm working on at the moment, and I would like to discuss further about this with you. Thank you. So this will be all about adult neurogenesis, and we will be meeting later in our lecture time.